Let's take a look at how microorganisms regulate their gene expression. That is going to be the topic of Lecture 18 in Bio 120. So most microbes must respond rapidly to changes in their environment. Processes no longer needed need to be shut off. Processes that are needed needs to be initiated for the microorganism to be able to live. So microbes efficiently direct activities by maintaining a really tight control over the gene product. For example, the downregulation of lactose catabolism enzymes when lactose is absent, and only when lactose is present, those enzymes will be then expressed. So let's take a look at how some of the regulation is acting across different processes. As you are aware, you have transcription, which is the initiation of gene expression from the DNA to make an RNA molecule. So you're going to have a really good way to control gene expression by controlling the rate in which the RNA molecule is made. You can also have control of gene expression by altering translation. So how is that mRNA now being read by the ribosomes and that protein being made? Last, you have mechanisms to controlling enzyme activity. You're going to have enzymes that get degraded. You're going to have allosteric feedback inhibition. You can have protein-protein interactions that are going to allow an enzyme to be either on or off, or you can covalently modify that enzyme, for example by phosphorylation, to activate that enzyme or inactivate it. So let's take a look at some of these potential mechanisms in which the microorganisms are going to regulate their gene expression. Now, we can look also that regulation is acting across different temporal scales. You can, as we discussed before, control transcription, so no mRNA molecules will be made. You can also control translation, so the enzyme will not be made. Now you can control enzyme activity, so the product of the enzyme no longer is made when the enzyme is inhibited. Otherwise, you can have a situation of no control when an enzyme that has been made, it's now able to take a substrate and create a product. So the regulation of enzyme activity is usually typically faster than the regulation of enzyme synthesis. It is easier to turn on or off an enzyme that is already made than to spend the time transcribing and translating those genes. So let's take a look at two examples of post-translational regulation that it happens after the protein is being made. For example, you can have proteolytic cleavage. An enzyme could be made as a pre-protein. For example, DNA gyrase, it's made as a protein that contains an intane. An intane is the protein equivalent of an intron. That intane needs to be removed, and once the intane is removed, you can have now the active subunit. The other thing that can happen is allosteric inhibition. For example, feedback inhibition that is going to be negative. As shown over here in this diagram from figure 7.34, you can start with a substrate. That substrate is going to be catalyzed by enzyme A to intermediate 1. Enzyme B will catalyze it to intermediate 2, enzyme C to intermediate 3, and enzyme D to the end product. Now, that end product can bind to enzyme A and by allosteric inhibition prevent the activity of that enzyme and therefore inhibiting the pathway that created it. So that feedback inhibition of the first enzyme is going to shut down the entire pathway. You can then also have degradation of the enzyme, or you can have a chemical modification of that protein, for example, as we mentioned, by phosphorylating it. As we learn in Bio 110, you can phosphorylate proteins to either turn them on or turn them off. So let's take a look at transcriptional control. You're going to have proteins that are able to bind DNA, and they're going to bind to regulatory sequences in the promoter. Those DNA binding proteins are called transcription factors. The transcription factors can bind DNA in a sequence-specific fashion. They have a target sequence that they recognize, and only if a promoter has that sequence would that transcription factor then bind. So it's only going to be found usually on the major groove, which is the most exposed area of the DNA molecule. So here's an example of a transcription factor. Usually a transcription factor are always found as dimers because the areas in which they bind are always found as inverted repeats. So here in this example, we have two pairs of inverted repeats, one blue and one lilac. And as you can see, now you have the opposite sequence in the right-hand side going lilac and then blue. each DNA domain that is going to fit in the major group, it's going to bind in that area, recognizing that sequence of DNA that are inverted repeats. Transcription factors now can come in multiple different types. I'm going to introduce you to 3 different structural types of transcription factors. You have the helix turn helix transcription factor, which is usually involved about some kind of alpha helix, a loop that is going to be the turn, and then another helix. One of them is going to be a stabilizing helix, and the other one a recognition helix that will recognize the DNA sequence to bind. Otherwise are considered zinc fingers, and the zinc fingers basically are transcription factors who has a regulatory recognition helix, but also are able to bind zinc through cysteine and histidine molecules. Last but not least, we have the losing zipper. The losing zipper is a transcription factor that has the recognition helices binding to the major groove, but the stabilizing helices are kept together by hydrophobic interactions between losing amino acids. So when we take a look at how, for example, a helix turn helix binds to DNA, you can appreciate here that the recognition domain is binding in the major groove. Now, on the image of the right, from figure 7.4, you can appreciate that this transcription factor is a dimer. It contains 2 subunits, one yellow and one orange. Sometimes the subunits are the same, other ones they may be different. But as you can appreciate, it is binding over here to the major groove. The transcription factor is not interacting really with the minor groove of DNA. Now let's take a look at some of the transcription regulation that is happening on the cell. And the first one that I want to introduce to you is regulation by repression. So regulation by repression can be observed and exemplified by looking at the arginine operon, which control enzymes involved in arginine biosynthesis. Here in this graph on the left, what we have are the relative increase of 3 different parameters: cell numbers in red, total protein in black, and the amount of arginine biosynthesis proteins in blue. So as you see, when the cell number and the total protein are growing at the beginning of our culture, the amount of arginine biosynthesis enzymes are also going up until arginine is finally made or added into the culture. At that point, there is no longer a relative increase in the amount of arginine biosynthesis enzymes. They actually plateau. So how is this system working? So the system for arginine biosynthesis contains an operon, as you know, that has a promoter. Here, the ARG promoter. Next to the ARG promoter is the ARG operator, and 3 genes, ARGC, B, and H, which are required for arginine biosynthesis. You also have a protein called the arginine repressor. In the absence of arginine, the arginine repressor doesn't bind to the arginine operator, and transcription proceeds forward. However, when sufficient arginine is present in the cell, arginine works as a co-repressor, binding to the repressor, inducing a conformational change that now is going to allow it to bind to the operator. At that point, transcription is blocked, and you do not have more transcription of the enzymes that are going to be required for arginine biosynthesis. Another type of regulation is regulation by derepression, and that is going to be exemplified by the lactose operon, which you already have learned quite a bit in other classes, but today what I want to show you are the multiple ways in which the lac operon is repressed. So here again we are going to look at an example of this. We also can call it induction, and here again in red we have the cell numbers, in black we have the total protein, and in blue we have the amount of beta-galactosidase enzyme. So we're going to look at the relative increase on cell number, total protein, and beta-galactosidase as opposed to time. Now, as you can appreciate, there is no increase in the amount of beta-galactosidase in the culture system until lactose is added. At that point, you have a sharp increase in the amount of beta-galactosidase present. So let's take a look at how the system works. So in this case, the lac promoter contains a regulatory sequence that is next to it called the lac operator. And you have 3 genes involved in this operon: lac Z, lac Y, and lac A. You also have a product of a 4th gene. That's the LAC-I gene that creates the LAC repressor, shown over here. Now, the LAC repressor is able to bind to the operator region and inhibit transcription. So, under steady-state conditions, the LAC operon is shut down in the absence of lactose. But when lactose is present, it behaves as an inducer. It binds to the repressor and is able to induce a conformational change on the repressor that no longer allows it to bind to the LAC operator. Now that the repressor is not present any longer at the operator, the RNA polymerase is able to transcribe the entire operon and induce production of beta-galactosidase and all the other components of the repressor. Just to remind you, beta-galactosidase is an enzyme that is going to hydrolyze lactose into glucose and galactose. So let's take a look at a process called catabolic repression, which is also understood as the glucose effect. This is a way in which lactose operon is also repressed and controlled by the presence of glucose. As we have learned in class, glucose tends to be a preferred carbon source for many cells. And glucose is going to induce a repression in the synthesis of beta-galactosidase. So those enzymes that are going to be made to synthesize lactose are not made if glucose is present. So let's take an example of a culture that has both glucose and lactose together. So what we can see in this graph, it's two different components. So the relative cell density in red and in green, red showing the growth during the glucose phase and green showing the growth during the lactose phase. In blue, we also have the relative level of beta-galactosidase. So what you can observe from this graph is that in the presence of both glucose and lactose, the initial growth curve that is happening exponentially is based on glucose. Why do we know that? Because the enzymes required for lactose biosynthesis, i.e. beta-galactosidase, are not present. It is only when glucose is exhausted, here in about 2 hours, that we begin to see the synthesis of beta-galactosidase. And at that point, we begin a new growth phase, now using beta-galactosidase to break down lactose into glucose and galactose. And as you can appreciate, the slope of that growth rate is also a little bit more shallow than the growth rate of cells growing in glucose. So glucose is providing a much better preferred energy source than lactose. The cells are going to grow a little bit slower in lactose as a carbon source. So when we're looking at this kind of dioxic growth, when we have glucose and lactose, we can see that you have two exponential growth phases, the one that is very steep for glucose and the one that is a little bit more shallow for lactose. So how is this mediated? Well, this is mediated by the relative amount of cyclic AMP, and cyclic AMP has an inverse relationship with the amount of glucose. So in conditions of high glucose, you're going to have low cyclic AMP. In conditions of low glucose, you're going to have high cyclic AMP. Now, cyclic AMP is a messenger that can bind to the cyclic AMP protein, or CRP. We also know that protein as the Catabolic Activator protein, or CAP. And in here in this image, what you can appreciate is the way in which cyclic AMP is made. So you can see that cyclic AMP is going to have the adenine, the ribose, and then the phosphate bond that is happening between the 5' carbon and the 3' carbon. Now let's take a look at how this amount of cyclic AMP helps to positively regulate lactose operon. And that is happening because of another site in the lactose operon called the CAP binding site. The CAP binding site, it's the site where the CAP protein is going to bind. So cyclic AMP, as we discussed, binds to the Catabolic Activator protein, or CAP, and once that protein has bound to cyclic AMP, it can dimerize and then bind directly to the CAP binding site. So the CAP is a sequence-specific DNA binding protein that binds DNA when the CAP protein has bound to cyclic AMP. And it is present in many operons. It is believed to be found in over 100 different catabolite-sensitive operons, and therefore it's able to increase the amount of expression of those operons in the absence of glucose. So in the presence of cyclic AMP, the cap cyclic AMP dimer can then bind to specific sites in the DNA in order to promote the binding of the cap and then increase the function of the RNA polymerase. So how is that accomplished? Well, the cap cyclic AMP dimer binds to a single DNA site upstream of the lactose promoter. So I remember that's the actin binding site to it. Now, cap is interesting because it is able to interact with the alpha CDT, basically the alpha protein C-terminal domain of the RNA polymerase. Remember that the RNA polymerase have an alpha, a beta prime, and the sigma factor as part of the proteins that make it. So the cyclic AMP cap protein interacts by binding to the carboxyl terminus of the alpha domain, shown here with the C and looking like the legs of a grasshopper. Yeah, don't hate me because to me it looks like a grasshopper, but anyway, it looks like the legs of a grasshopper. So it's binding to the C area. Now, once it binds to that C area, it's able to facilitate the binding of the RNA polymerase to the RNA promoter, helping the sigma factor recognize the -10 and -35 region of the promoter. So it's going to help stabilize that structure. And once the RNA polymerase is bound to the promoter, it can initiate transcription and cap is no longer needed. So cap helps to stabilize the RNA polymerase binding to the promoter. So let's take a look at the overall regulation for the LAC system. And we can think about it as having two different levels of control. The first one is the repression of the promoter by the binding of the LAC repressor. So the product of the LAC-I gene is going to bind to the operator, and that is going to inhibit transcription. And when lactose is present, lactose is going to be able to bind to the active repressor and inhibits its binding to the operator. So you have one level of regulation right there. The other level, it's the activation via cyclic AMP and cap. As you know, as low levels of glucose, you're going to have high amounts of cyclic AMP. That is going to then allow the cyclic AMP cap protein to form a complex that can then bind to the cap site. And that is going to then recruit the RNA polymerase to activate transcription. This is the information that is described here in this image. Let's take a look now and examine regulation by induction. And here, as an example of this induction, we're going to have the maltose operon. The maltose operon has one promoter, it doesn't have an operator, and it has three genes, the MAL-E, MAL-F, and MAL-G. Now, it has an activator binding site upstream of the MAL promoter. And you also have the product of a maltose activating protein, shown here in yellow. In the absence of maltose, the maltose activator protein does not bind to the activator binding site, and therefore it cannot help the RNA polymerase to bind to the MAL promoter, and therefore there's going to be no transcription. However, when maltose is present, maltose functions as an inducer, so it's able to bind to the maltose activating protein, which can now recognize the activator binding site, and therefore it helps recruit the RNA polymerase to the MAL promoter, initiating transcription of the maltose operon. So it's a positive control mechanism mediated by maltose. So it requires the maltose activating protein, and it requires the inducer that is going to be maltose. Some operons, you're going to see that the activator binding sites doesn't need to be directly upstream of the promoter. Some of them are at longer distance away from the promoter, and the reason that they are working is because as the DNA molecule is coiling, that activator is able then to come to close proximity once the activator protein and the inducer are present to bring that close to the promoter and the RNA polymerase. So therefore it's able to help activate transcription from a distance. So let's take a look at a fourth different way in which certain operons are controlled, and that is the process of attenuation. So for attenuation, we're going to use an example of the tryptophan operon. The tryptophan operon is a normal garden variety operon that contains a promoter, it contains an operator, and it has also a leader regulatory element. Now, Repression controls this operon, so it's a weak negative regulation. So in the presence of tryptophan, tryptophan repressor binds to the operator. Kind of like the same way that arginine represses. So you have to have tryptophan present to help the tryp repressor bind to this operator and inhibit transcription. But there is a second interesting way in which regulation exists, and that regulation happens by attenuation. Attenuation requires a leader sequence. So here in this image, you can have the promoter, the operator that is controlling negative regulator, but you also have a leader sequence. This leader sequence is a sequence of RNA that encodes for 14 codons for 14 different amino acids. But in highlight here in red, I want to point out to you that you have consecutively 2 tryptophan amino acids present in the leader. Then the structural genes are going to include the TRPE, TRPD, TRPC, B, and TRPA genes. Now, that leader encodes an RNA, and that RNA eventually will be translated into a peptide that has 2 tryptophans. When tryptophan is at high levels, the leader peptide is going to be synthesized because the ribosome is able to put the amino acids directly here from the tRNAs. When tryptophan levels are low, the ribosome is going to stall when it finds those strip codons, and therefore the peptide sequence is not going to be synthesized. Let's take a look in more detail to see how that occurs in the bacterial cell. So let's start by looking at these 2 situations. The first thing that I want to bring up to you is that the mRNA contains 4 regions. We're going to have region 1, which is going to be the leader, region 2, region 3, and region 4. What we're going to see is that the leader peptide is encoded in the RNA region 1, and that is where you can find the tryptophan codons present. When tryptophan is at low levels, as shown in the image to the right, the ribosome is going to begin translating the mRNA that comes. But when it gets to those 2 tryptophan codons, it's going to stall. There is no tRNA with tryptophan because the amount of tryptophan is very limited. That ribosome then is going to stall at position number 1. The RNA polymerase is going to continue synthesizing the mRNA. And now you're going to have sufficient mRNA to form a stem loop between regions 2 and regions 3. That stem loop is going to be formed in that way, and it's going to prevent the ribosome from going forward, but it's going to allow the RNA polymerase to continue with transcription, and therefore you're going to transcribe the structural genes that are required to make tryptophan. On the other hand, if the amount of tryptophan is high, you're going to begin to translate the leader sequence, shown over here. The ribosome is going to bypass and add amino acids at those 2 tryptophan codons, and therefore eventually is going to cover region 2. Now, instead of making a loop between region 2 and region 3, the RNA is going to make a loop between region 3 and region 4. That loop is going to be a hairpin termination loop. And as you remember, the hairpin termination loops are going to help destabilize the RNA polymerase and stop transcription. And therefore the RNA polymerase is going to fall off out of the DNA, and that is going to prevent the tryptophan structural genes from being transcribed. So the key process that ensures that attenuation works is the fact that in bacteria, transcription and translation are coupled. Therefore, they're happening inside the cytoplasm. In eukaryotes, as you know, transcription occurs in the nucleus and translation occurs in the cytoplasm. So the process of attenuation will not work in a eukaryotic cell. This image here is showing a better representation of attenuation. The upper part is showing the case with high-level tryptophan, in which you have the trip codons, and therefore the ribosome is able to add those amino acids to the sequence, making the sequence peptide, and continue blocking the area of the mRNA called loop 2. That is going to make this stem loop that is a transcriptional terminator, and the RNA polymerase will fall off the DNA. On the other hand, when you have low levels of tryptophan, the amino acids are no longer there, and the ribosome is going to stall at position 1 because it cannot add an amino acid to the sequence because it cannot read them. There is no tRNA with tryptophan attached to it, and therefore the amino acid synthesis will halt and the ribosome will stall. Now, the stem loop is going to be formed between region 2 and region 3. That is an alternate stem loop which allows transcription to occur. And now the RNA polymerase is going to be able then to transcribe the genes that are required for tryptophan biosynthesis. What we're going to see is that other amino acids are going to have operons that also use attenuation. So we already talked about tryptophan, but you're going to see that threonine is another operon that is going to have attenuation. Notice the long chain of threonines present here in the leader peptide that contains about 1, 2, 3, 4, 5, 6, 7, 8 different sites for threonine addition. So if threonine is not made, the ribosome will stall in this area of the leader mRNA. Histidine has a region with 7 histidine amino acids, and therefore, as the ribosome is trying to translate the mRNA, once it gets to this area with 7 histidines, it's not going to be able to add them, and therefore it stalls. The last one is going to be phenylalanine. Phenylalanine has 2 regions on its leader peptide that contain phenylalanine. 2 regions with 3 amino acids and 1 with a single one. So once the ribosome reaches this area and is trying to add the amino acid phenylalanine, if they are not present, the ribosome will stall, allowing for the transcription of the phenylalanine structural genes to occur. One other way that you're going to discuss more in detail in the next lecture is going to be Corrin Sensing. And just to give you an idea, I'm going to mention here, and Corrin Sensing is a mechanism used by bacteria to count how many organisms they are present in an environment. And it requires the presence of an autoinducer, an acyl homoserine lactone, or AHL, that is specific for microorganisms of the same species. So here in the image, we have other cells of the same species which are releasing AHL, and a bacterial cell of the same species. Since the concentration of AHL is going to be high on the outside, it's going to diffuse passively inside the cell, activating an activator protein that is going to result in the activation of Corrin-specific proteins. One of them is going to be the AHL synthase, which is going to result then in the production of more AHL, which is going to result in the production of more AHL. Vibrio fischeri, it's a marine gram-negative microorganism whose genes for bioluminescence are controlled under Corrin Sensing, and that is the image that you see here in the right. We're going to spend some more time in the next lecture looking at that for your enjoyment. The last system that I want to review with you is the 2-component regulatory system. And this system, as its name implies, it has 2 components. One is going to be a sensor kinase. The sensor kinase is going to be sensing the environment, and when a signal comes in, that sensor kinase is going to undergo autophosphorylation. So once it's autophosphorylated, it's going to now be able to transfer that phosphate to a response regulator that is going to activate it. That is the second component, the response regulator. And the response regulator comes in many different shapes. We're going to see that some of them are going to be DNA binding proteins. So once they are phosphorylated, they will be able then to bind to DNA and activate transcription. So upon activation, they work as transcription factors for many genes. So some other response regulators regulate activity in other proteins when they're active. So let me give you 2 different examples of this process. Here is an image of the general concept of the 2-component regulatory system. Here it's the cytoplasmic membrane where you have the presence of the sensor kinase. The sensor kinase tend to be histidine kinases. So once the environmental signal binds to that sensor kinase, the sensor kinase is going to autophosphorylate its histidine. Now that it is phosphorylated, it is able to pass that phosphate and phosphorylate a response regulator, which is now shown here phosphorylated. Now, In this case, it's an activator molecule that is able to bind to the operator in the operon, and in this case, block RNA polymerase from transcribing. In other cases, the response regulator can activate RNA polymerase and help activate transcription. So depending on the system, the response regulators are going to have different kinds of effects. For example, E. coli is able to respond really quickly to changes in osmotic pressure, and it contains two different osmotic regulatory porins. One called the low osmolarity porin, shown here in yellow, that is the protein encoded by the gene OMPF, and another one called the high osmolarity porin, encoded by the gene OMPC. Now, that system allows for the passage of solutes because the OMPF low osmolarity porin is a lot bigger than the OMPC porin, and therefore a lot more molecules can pass through it. Now, in the cell, you have the NVZ kinase. The NVC kinase, it's the sensory kinase that is going to sense changes in membrane surface tension when the osmolarity changes. Once that signal has passed, it's going to autophosphorylate its histidine, and now those variations in membrane surface tension are going to induce that conformational change that is going to allow the phosphorylated NVZ to pass that phosphate to the response regulator, which is the protein OMPR. OMPR is going to then be phosphorylated by NVC. And during low osmolarity conditions, OMPR is going to bind to the promoter of the OMPF porin. Now, the OMPF promoter has 3 high affinity OMPR sites and 1 low affinity site. So during low osmolarity conditions, the enzyme NVC doesn't get phosphorylated very often, and therefore you have very little amount of phosphorylated OMPR, which can bind to the very high affinity OMPR phosphorylation sites on the OMPF promoter. Now, when the osmolarity goes high, that is going to turn on NVZ to be phosphorylated very rapidly and now increase the amount of OMPR that is phosphorylated. And that is going to now cause the OMPR to bind again to the OMPF promoter, but now is binding to the high affinity sites and also to the low affinity sites. And that is going to turn that promoter off. So binding to the 3 high affinity sites at low concentration turns the promoter on, but binding to the high affinity OMPR receptor and the low affinity one is going to turn that promoter off. Therefore, at high osmolarity, you shut down the OMPF protein. However, the OMPC promoter has 3 low affinity phosphorylation sites, and therefore, at high amount of OMPR resulting from high osmolarity, it's going to allow to the OMPR phosphorylated to bind to the promoter of the OMPC gene now, and therefore that is the only protein that is made. So the differential binding of the OMPR with phosphate to the promoters of the OMPF and OMPC result in the either turning on the promoter at low osmolarity concentrations or turning the promoter off in high osmolarity concentrations. Let's look at another example in which the two-component system is used to control cell behavior, and that is going to be the run and tumble behavior in chemotaxis. So bacteria, as you know, are making temporal comparisons. They're trying to determine in which direction they are going. So they're going to have a response to those spatial gradients. So a compare of the environment of the present with the environment of the past is going to allow the microorganism to eventually reach the source of an attractant or run away from a repellent. And that is going to induce changes in the run frequency, which are then intermitted by tumble behavior. that is mediated by the chemotaxis mechanism. So we have in this system a chemotaxis mechanism involving a methyl-accepting chemotaxis protein, or Ncp, and a kinase, the key A molecule. And the response regulator is going to be two, key Y and key B. Now in this case, when an attractant is bound to the receptor, the key A is non-phosphorylated, and that is going to allow for the microorganism to have counterclockwise flagellar movement that are going to induce a run. But when the attractant is not present, that is going to allow the key A to be phosphorylated. Phosphorylated key A is able now to transfer its phosphate to key Y, and phosphorylated key Y is going to be able to bind to the flea proteins in the flagellum and induce a change in the rotation to counterclockwise and inducing a tumble. So the response to signal and flagellar control is mediated by the phosphorylation of key A and subsequently key Y. But there's a second level of control existing called adaptation, and the adaptation is an idea in which the amount of signal is regulated, and this is mediated via the methylation of the Ncp proteins. Methylation of the Ncp proteins happens by the enzyme key R, and key R is going to add methyls to the receptor. The receptor, once it's methylated, it's less responsive to regulation. Now, when the receptor is not bound to the attractant, key A is going to be phosphorylated, and now key A is going to be able to pass its phosphate to another adapter molecule, key B. Key B is a methyl esterase and is going to mediate the removal of the methyl groups from the receptor. So how does this work? Let's check out the response. So the methyl-accepting chemotaxis proteins is going to detect the presence of an attractant or a repellent. Now, key A is the sensor kinase. The attractants are going to decrease the rate of autophosphorylation of key A, versus repellents are going to increase the rate of autophosphorylation on key A. The autophosphorylated key A is going to then transfer its phosphates to the response regulators key Y or key B. Key A is a histidine kinase, and therefore it is not a kinase that is actively able to transfer its phosphoryl group. The response regulator key B sequesters that phosphate group from key A and autophosphorylates itself. Key B phosphate acts as a methyl esterase and is able to remove the methyl groups from the Ncp receptor. On the other hand, key Y phosphate is going to interact with the flagellar motor flea M, and that is going to cause the counterclockwise rotation that really results in a tumble. On the other hand, you have a phosphatase called key Z, and key Z is going to dephosphorylate key Y phosphate, allowing now the flagellum to return to the counterclockwise motion and have a run. So let's check the response again. The key R is going to continually add methyl groups to the Ncp protein at glutamate residues, and is adding that constantly at a slow rate. The phosphorylated key B is a methyl esterase and is able then to remove those methyl groups from Ncp. So key B phosphate and key R are working antagonistically to one another. One adds methyl groups and the other one removes them. Now the level of methylation of the Ncp is going to affect its conformation and control the adaptation to a sensory signal. If the level of attractant remains high, the level of key A phosphate and therefore key Y phosphate and key B phosphate is going to remain low. The cell will swim smoothly and the level of methylation of the MCPs will increase because you have a low amount of key B phosphate that is not then able to demethylate the MCP. MCPs no longer respond to the attractant when they are completely fully methylated. Therefore, even though the level of attractant may remain high, the level of key A phosphate and key B phosphate increases and the cell now begins to tumble. However, now the NCP can be demethylated by the key B phosphate, and when this happens, the receptor once again can respond to attractants and allow the cell to run. So regulation of chemotaxis is complex and is involving a response and an adaptation to that response. The response arises from the combination of A, the control of key A phosphorylation by the attractant or the repellent levels, the control of the rotation of the flagellum exerted by the key Y phosphate, and the feedback regulatory circuit involving the key R, key B phosphate, and the variation of the methylation amount on the Ncp. This regulation then allows the bacteria to remember a chemical concentration from this recent past, from a few seconds, for example, and be able to compare them to those that is currently experiencing and then quote-unquote know whether it is traveling up or down a gradient of an attractant or a repellent. With that, we'll stop the lecture. Remember, we'll have a quiz about this on Tuesday, and I wish you a very happy beginning of the week. Bye-bye.